Imagine you are looking at a highly detailed satellite map of a massive, sprawling city. You're trying to understand how the city flows, right?
You want to see where the high-density downtown area ends.
Exactly. Or where the quiet, winding residential neighborhoods begin. But instead of tracing the natural, organic borders of, like, rivers or highways or parks, you just take a giant ruler and slap a rigid, perfectly square grid over the entire map.
Which is a terrible idea because suddenly you've accidentally sliced a massive skyscraper right in half.
Right. You've grouped half of a serene, quiet park with a noisy, polluting industrial plant just because they happen to fall inside the same arbitrary square. It makes absolutely no sense.
No, it really doesn't.
Well, it turns out this is exactly what current artificial intelligence has been doing to human tissue when trying to diagnose cancer.
It is, and the consequences of forcing that rigid grid onto organic biology are profound. In computational pathology, which is essentially the field of using AI to analyze tissue samples to find things like tumors, we're dealing with highly complex natural structures.
Very fluid, organic stuff.
Right. The exact irregular shape of a tumor is what we call the region of interest, or the ROI. That jagged, weirdly shaped border is absolutely crucial for a doctor to understand the disease.
Because tumors don't grow in perfect squares.
Exactly. So when an AI just blindly chops a tissue slide into arbitrary squares, it completely misses the coherent architecture of the tissue. That limits both the clinical relevance of the AI's predictions and, more importantly, our ability to actually interpret why the AI made a specific diagnosis in the first place.
Okay, let's unpack this, because this completely shifts how we think about medical AI. Our deep dive today is into a groundbreaking paper detailing a new foundation model called CARE.
Which stands for Cross-modal Adaptive Region Encoder.
Right. And our mission today is to unpack exactly how CARE stops arbitrarily chopping up these critical medical images and instead learns to actually see the fluid, organic boundaries of human tissue. And the craziest part, it learns to draw these boundaries guided by the tissue's very own invisible DNA and protein blueprints.
To really appreciate the leap forward that CARE represents, we first need to look at the flawed status quo. We have to understand what the AI is actually looking at. In pathology, doctors use what are called whole slide images, or WSIs. And these are gigapixel images. Gigapixels. So we are talking over 1 billion pixels per image, right? Scanned at a massive 20x magnification.
Yeah, so these aren't just your standard smartphone photos.
They're astronomically huge files.
Exactly. They are so massive that feeding an entire high-resolution image into a neural network end-to-end all at once is computationally impossible.
The server would just completely crash.
It would. So to deal with the sheer size, the current standard practice is to use what researchers call patch chunks. You essentially tile the image into thousands of tiny fixed-size squares.
This is our rigid city grid from the map analogy earlier.
Right. But there's actually a brilliant analogy in the source text that compares this to natural language processing, or NLP. In language models, the AI has to break text down into pieces to process it.
Sure, like tokens. Yeah. If you use rigid patch chunks in pathology, it's the exact equivalent of forcing an AI to read a novel character by character. It's incredibly fine-grained, but completely myopic. You see the letter T, the letter U, the letter M, but you completely miss the word tumor.
Oh, wow, that makes perfect sense. You totally lose the forest for the trees. And, I know some previous models realized this was a problem, so they tried to group these little patches together into what they called regular region chunks.
Right, but they just use bigger, fixed grids based on rigid rules.
Yeah.
Yeah.
In our NLP analogy, that's like arbitrarily chopping a sentence every, say, 5 characters.
Oh, yeah.
So you'd read fissy, sussin, tense. It completely destroys the semantic meaning of the words.
It absolutely shatters the context.
Yeah.
And that lack of boundary respect is entirely missing, even in the most massive state-of-the-art foundation models we have today, like Chief or Gigapath or Titan.
Which are huge names in the field.
They are, but the issue is that those models rely on architectures originally built for analyzing regular, everyday photos like pictures of dogs or cars. They lack any explicit concept of a region of interest.
So to compensate, they're forced to rely on global self-attention across the entire whole slide.
Precisely.
Meaning the AI is desperately trying to calculate the mathematical relationship between thousands and thousands of tiny disconnected square patches all at once, from completely opposite sides of the tissue sample.
It's an incredibly inefficient way to learn. CARE, on the other hand, introduces adaptive region chunks. Instead of arbitrary letters, these act like actual word-level tokens. They respect the semantic biological boundaries of the tissue sentence.
If human pathologists naturally zoom in on a specific, weirdly shaped region of interest, why did it take AI so long to ditch the rigid squares?
What's fascinating here is that traditional foundation models, because they lack that explicit ROI concept entirely, make the aggregator incredibly hard to learn. They just didn't have the architecture to let the image organically group itself.
Okay, so let's do a quick recap here. Bullet point 1, whole slide images are just too big to process whole. Bullet 2, old AI uses rigid patches, which is like reading individual letters. And bullet 3, care uses adaptive regions, acting like meaningful words.
That's the perfect summary.
And think about how you read a book, you. Do you read letter by letter, or do you absorb whole words and phrases? Why should we expect an AI diagnosing something as serious as cancer to do it any differently?
We really shouldn't. And that brings us to the actual mechanism of how this is achieved. Because, it's one thing to say we need organic boundaries, but it's another to mathematically execute it.
Right. How does care actually group these rigid patches together organically without a human holding its hand?
It does this using a brilliant module called the Adaptive Region Generator, or the ARG.
I love this part. So let's get into the mechanics of the ARG. We start with our massive WSI and we extract all our tiny square patches.
Right, and to do that extraction, they use a specialized patch encoder called CONCH version 1.5. You can think of TONCH as a hyper-focused mini-summarizer.
Okay.
It looks at one tiny square and extracts a highly detailed feature representation, which they call F sub I, of just that specific area. Then, the slide is initially divided into a set number of non-overlapping subregions, just as a starting point. Let's call that number M.
But here's where the magic happens, right? Because historically, a patch was permanently locked into whatever arbitrary starting square it was dropped into.
Exactly.
But with the ARG, the patch actually gets to choose its optimal home.
It does. It calculates what's called a soft inclusion score. The patch looks around its immediate neighborhood and measures its spatial distance to the center of nearby subregions. It's primarily looking for physical proximity.
I picture this almost like a magnetic attraction. The patch is physically feeling the pull of the regions that are closest to it. So it takes that magnetic pull and shortlists its top 3 candidate subregions.
Once it has that top 3 shortlist, the patch runs a quick calculation to find its regional selection score. Think of it as a biological compatibility test using a softmax function.
So it's basically asking, okay, of these 3 nearby regions, which one actually looks like me in terms of cellular structure and semantic affinity?
Exactly. It assigns itself to the absolute best fit among the 3. And as thousands of patches do this simultaneously, they naturally create a new, fluid, irregular boundary. This is your adaptive region.
And once that beautiful irregular region is formed, the AI does something really clever with its attention mechanism. They use something called RRSSA adaptive region self-attention.
Yes, the RSA module.
They basically drop a learnable token into this new region, which acts almost like a team captain. From that point on, the self-attention mechanism, the part of the AI that connects the dots, is restricted to only look within this newly formed, biologically accurate region.
By restricting the attention locally, you immediately eliminate all the noisy, irrelevant data from the complete opposite side of the tissue sample. It saves massive amounts of computational power.
Wait, I need to stop you there and push back on this a little bit.
Sure.
If we are giving these patches the freedom to choose their own borders and draw their own shapes, aren't we risking massive hallucinations? Like, what stops the AI from being incredibly lazy, looking at the entire slide, and just glomming all the squares together into one giant, useless mega region just to make the math easier?
That is the exact trap the AI would fall into without a specific safeguard.
Okay, so there is a safeguard.
Yes. To prevent that lazy clumping, the researchers built in the region structuring loss, or RSL. You can think of RSL as a penalty function built directly into the training process.
Interesting.
It constantly monitors the expected rank distribution of the patch's choices.
Meaning it's watching how they choose?
Yes. It essentially forces the AI to maintain diversity. If the algorithm starts getting lazy, if patches just blindly pick the biggest, most dominant region every single time to save effort, the RSL penalizes the model.
So it keeps it honest.
Exactly, and ensures that the natural, complex, heterogeneous landscape of the actual tissue is preserved, rather than collapsing into a single, useless blob.
All right, time for another quick recap. Bullet 1, the ARG groups patches based on spatial distance and semantic similarity. Bullet 2, patches get to choose from a top 3 candidate list. And bullet 3, That RSL penalty prevents the AI from just collapsing everything into a single blob.
Spot on.
If you were designing an algorithm to group your daily tasks, would you force them into rigid one-hour blocks? Or let them organically group together based on your fluctuating energy levels? It just makes more sense to be adaptable.
It really is a huge leap in efficiency. But that organic grouping raises another massive hurdle.
What's that?
Well, grouping visually similar patches is a great start, but how does care actually know what a biologically meaningful group looks like? A cluster of cells might look identical visually, but have completely different underlying functions or mutations.
Ah, right. Looks can be deceiving.
Exactly. To solve this, care uses a two-stage molecular pre-training pipeline, and this is where it really separates itself from older models.
Okay, so we have the shape, but now we need the function. Stage 1 of this pipeline is unimodal IBRT pre-training. They use a massive data set of over 34,000 whole slide images.
Which is huge.
But we already established earlier that gigapixel images are absolute memory hogs. How do they train the AI on 34,000 of them without the servers literally catching fire?
That's where they bring in a technique called DBS-CAN clustering. It's an algorithm that intelligently breaks the massive slide into smaller, manageable chunks, what they call sub-WSIs.
Okay, so breaking it down.
Right. And they cap them at a maximum of 360 patches. This prevents the out-of-memory crashes while still preserving enough local context for the AI to learn.
Got it. And because they are using pre-extracted features rather than raw pixels, they can't do the standard parlor tricks of image augmentation like, flipping or rotating the image to train the AI.
No, they can't.
Instead, they mathematically tweak the brightness or contrast in the feature space itself using frozen feature augmentation. But the really critical part of stage 1 is that when they test the AI, when they do masked patch modeling and hide a piece of the image to ask the AI to guess what's missing, they restrict this guessing game strictly within the boundaries of the adaptive region they just built.
That is key. That restriction prevents cross-region noise from confusing the model. It learns the visual structure perfectly. But stage 2 is the true secret sauce.
Yeah.
Cross-modal alignment.
Right.
This is where they pair those visual features with the actual molecular data. They are feeding the AI the invisible blueprints of the tissue.
Here's where it gets really interesting. They do this using an info-NC loss function, which sounds complicated, but conceptually it just mathematically pulls matching visual images and molecular profiles closer together while pushing mismatched ones apart. But the curriculum order they do this in is really specific. They align the visual images first with RNA profiles, about 13,000 pairs of data, and then after that, they align it with protein profiles, using about 8,000 pairs. Why that specific order? RNA first, then protein.
It's about moving from a general foundation to highly specific expertise. The RNA data, which they initialize using a model called SCGPT, is broad.
Okay.
It offers high coverage supervision across the whole slide. It gives the model a fantastic general biological foundation. Protein data, however, is incredibly dense and highly specific to distinct, localized cellular functions.
So you build the broad, sturdy foundation with RNA, and then you sharpen the fine, hyperlocal details with the protein data. It's like teaching someone to identify a specific type of cake purely by showing them the chemical breakdown of the ingredients, right?
If we connect this to the bigger picture, this curriculum sharpens the region boundaries immensely. The region embeddings are no longer just visual textures. They directly reflect the underlying biology and gene expression.
Let's do our 15-minute recap here. Bullet 1, Stage 1 uses IBET learning with DBS-CAN chunking for memory efficiency. Bullet 2, Stage 2 aligns images with RNA first, then protein. And bullet 3, this molecular data forces the visual regions to match real biological functions.
Perfect.
And for you listening, how often do you use two completely different senses like sight and sound to verify a single piece of complex information in your own life? That's exactly what CARE is doing with vision and molecular data.
And that leads us to the ultimate test. We have this brilliant organic architecture, and it's been cross-trained on pure molecular truth. The final step is seeing what happens when we unleash it on actual cancer prediction tasks.
Right, the benchmarks.
This is where the final piece of the puzzle, the SPF module, comes into play.
Right, SPF semantic and prior fusion. Now that the AI has drawn all these different fluid, irregular regions across the tissue, it needs to synthesize them. SPF is a lightweight module that pools all those regions together to create one final WSI embedding.
It does this by using gated attention, specifically tan and sigmoid functions, combined with a coverage prior. It evaluates all the adaptive regions it has created, assigns them a weight based on their importance to the diagnosis. And the single region that receives the absolute highest weight is crowned the explicit region of interest, the ROI.
It points directly to it and says, this is the most critical area.
Yes.
And we have to talk about the results because they are staggering. They tested care on 33 different downstream benchmarks. We're talking everything from basic morphology classification to predicting highly specific molecular mutations like TP53 or HR2, all the way to survival analysis, actually predicting patient outcomes.
And CARE achieved state-of-the-art results across the board. But what researchers are really marveling at is the data efficiency. CARE consistently beat out massive resource-heavy models like Titan and Prism.
Wow.
And it did this while using roughly 1/10 of the pre-training data that those massive models required.
So what does this all mean? It is the absolute definition of working smarter, not harder, because it's not wasting time looking at irrelevant, chopped-up background noise. It learns so much faster.
Exactly.
But the absolute coolest part of this paper for me was the visual proof. They included a case study on a specific cancer, CPTAC-HNSCC, that's head and neck squamous cell carcinoma.
Yes, the visual heat maps in that case study are the ultimate validation of this entire approach.
In this case study, human pathologists, actual expert doctors, looked at a slide and manually drew highly specific, jagged blue contours around areas of the tissue that showed nuclear atypia, which basically means weird, abnormal-looking nuclei, and numerous mitotic figures, which are cells caught right in the act of rapidly dividing.
Right, very complex areas.
They drew these incredibly complex, irregular borders by hand.
And when older AI models looked at that exact same slide, their attention heat maps were vague and messy. They just highlighted random, broad, blocky areas that bled way outside the actual tumor.
But CARE is different.
Entirely. When you look at CARE's heat map, specifically the version of CARE that was fully trained with the RNA data, it intensely highlighted the exact irregular, jagged shape that the human pathologist had drawn. It was a perfect match.
Which means we finally have an AI that sees the tumor the exact way a human expert sees the tumor.
This raises an important question about clinical deployment.
Yeah.
In the real world, doctors are highly skeptical of AI, and rightly so. They don't just want a black box AI spitting out a survival prediction with a percentage attached to it.
No, they demand interpretability.
They want the AI to show its work.
Yeah.
Because CARE's SPF module explicitly defines and highlights that ROI, the doctor can look at the exact, irregularly shaped cluster of cells the AI used to make its decision. It completely demystifies the AI's logic.
So recap time. Bullet 1, SPF aggregates regions and crowns 1 the ROI. Bullet 2, CARE beats massive models on 33 tasks with 90% less data. And bullet 3, visual heat maps perfectly match human pathologist annotations.
Exactly right.
Think about it. Do you trust an AI's answer more if it can explicitly highlight the exact scratch pad it used to get there? You are always going to trust the scratch pad more. So looking at the big picture, we have journeyed from an AI that stubbornly forced organic human tissue into rigid, arbitrary grids to a highly fluid, molecularly guided system.
It's a total paradigm shift.
CARE proves that by simply allowing an AI to adapt its borders, treating tissue structures like meaningful words in a sentence rather than random letters, we unlock incredible efficiency and deep clinical accuracy.
We are no longer fighting the natural biology of the tissue. We're finally letting the AI learn from it.
And it leaves us with such a fascinating concept to end on. CARE uses invisible data molecular RNA and protein profiles to fundamentally change how an AI interprets and sees physical, visual tissue structures.
It does.
If we can map the invisible molecular world onto physical medical images to accurately predict disease, well, what other invisible data sets are out there? What if, one day, we could map a patient's historical environmental exposures, like the air quality of the city they lived in, or even their lifetime stress and cortisol data, onto physical scans?
That would be incredible.
Could we teach an AI to see the ghosts of those invisible experiences imprinted in the tissue, predicting severe illnesses before the cells even begin to physically change shape? It's definitely something for you to mull over. Keep questioning the rigid, grid-like systems in your own life and your own work, because as CARE beautifully demonstrates, the most meaningful answers are almost always found in the organic, irregular spaces in between. Thanks for diving deep with us.